Chlorophyll fluorescence in plant phenotyping. Can we shorten the adaptation time to accelerate phenotyping processes?
Chlorophyll fluorescence is frequently used in plant phenotyping. However, standard chlorophyll fluorescence protocols frequently include quite long-term adaptation times, for instance dark adaptation or light adaptation, usually lasting several minutes. This slows down the phenotyping processes. The protocols have been developed for photosynthesis research, but do we need the long-term adaptation time in any case when it's about phenotyping, for instance to detect stress? I made some tests and figured out how far can we go to reduce the adaptation time and still see whether there is a difference between stressed and non-stressed plants in the fluorescent signal.
To try it out, I treated some plants with herbicides, because herbicides definitely make stress, and this stress is known to turn out in changes in chlorophyll fluorescence. I applied diurone, which blocks the electron transport in photosynthesis, glyphosate, that blocks the Shikimate pathway in amino acid synthesis, and pelargonic acid, which destroys the cell structures. I used the different herbicides because I wanted to evoke different effects in the plant that turn out into differences in the fluorescent signal. I incubated the leaves after the herbicide treatment for about 1 hour. At that time, no stress was visible at the leaves. Then I started testing chlorophyll fluorescence protocols. First, I did a standard protocol. I dark adapted the plants for about 20 minutes, then took a dark adapted chlorophyll fluorescence recording, light adapted for 5 minutes, and then took again a light adapted chlorophyll fluorescence assessment. The herbicide treatments led to changes in both the dark adapted and the light adapted chlorophyll fluorescence data. In the next step, I shortened down the dark and the light adaptation time to see whether still signatures of damage and stress could be seen in the chlorophyll fluorescence data that were acquired now not after the standard dark or light adapting time, but after a shortened adaptation time. I could shorten down the dark adaptation time to just 1 minute and the light adaptation time to 1.5 minutes, and still could detect the stress that was caused by the herbicide. So in case of those herbicide-treated leaves, even a very short dark and light adaptation time was sufficient to see very similar stress signatures as I could observe them after standard adaptation times. By the way, let's have a quick look what the herbicides did. Diurone blocks the electron transport, and we see a decrease in Fv over Fm as well as in light-adapted quantum yield. This means it damages the photosynthetic apparatus and impairs the photosynthesis. Glyphosate blocks amino acid synthesis. This is a long-term effect. In Fv over Fm, the glyphosate-treated leaf looks similar to the untreated leaf. In light-adapted quantum yield, the values even seem to increase a bit. Maybe the plant tries to respond to the stress and intensifies photosynthesis. Pelargonic acid just destroys the plant cells, and we see that in both light and dark adapted status, the quantum yield is decreased very strongly. So the tissue is essentially dead, even if the leaf is still looking green. So coming back to the initial question, is it possible to shorten down the adaptation time when you operate chlorophyll fluorescence in plant phenotyping? I would say yes. The demonstration case with the herbicide showed that even after strongly shortened adaptation time, stress detection and stress discrimination was possible. Can we generalize that? I can't say at the moment. At least it's worth a try. When you want to discriminate between stress factors and see whether plants are stressed via using chlorophyll fluorescence in plant phenotyping, you could go for a test experiment and see whether you can detect the stress with shortened adaptation time.